This is the chapter of microorganism and their use in biotechnology. Previously, we saw the general structure of bacteria, virus, and fungus, and now we have to see their application around us, how it can be beneficial for mankind. First, we also should be knowing the different diseases that are caused by these microorganisms. Influenza, smallpox, herpes, and measles are viral diseases, while cholera, syphilis, and TB are bacterial. Fungal diseases include ringworm and Achilles foot. The application in biotechnology includes use of viruses as vectors to transfer genetic material between cells of two species. Then we will see the formation of different products, for instance, yogurt and cheese, by using different bacteria. Then we will also see that how bacteria is helping us to treat sewage and what's the importance of the plasmid of the bacteria in genetic engineering. Fungus is used for the formation of cheese, while it is also leading to the production of bread and alcohol. Let's see the formation of yogurt first. First step includes the pasteurization of milk at around 85 to 95 degrees Celsius for about 15 to 30 minutes. Then the milk is incubated with bacteria for about 4 to 6 hours at 38 to 44 Celsius. Then we will add a microorganism, which is actually a bacteria called Streptococcus, which will remove the oxygen, creating anaerobic condition in the vessel or the container. We are creating anaerobic conditions here because the next microorganism which we are going to add is lactobacillus, which respires anaerobically, converting milk sugar lactose to lactic acid. This lactic acid form would cause curdling of milk protein casein, and this curd or yogurt is industrially then flavored and preservatives are added sometimes. Then it is cooled to around 4 to 5 degrees, packed, stored at 2 degrees Celsius. So this was the manufacturing of yogurt by using microorganism. Here two microorganisms are used, Streptococcus and Lactobacillus. Next product is bread. The flour that we use for bread formation contains starch, protein, and amylase enzyme, which is mixed with water to form dough. Yeast is mixed with the dough. Amylase would start to digest the starch to sugar. Lack of oxygen inside the dough causes the yeast to respire anaerobically, fermenting the sugar to alcohol and carbon dioxide. This alcohol formed would be evaporated because of the heat during the baking, while carbon dioxide would be used to raise the dough. The protein gluten forms starchy fibers which will trap these carbon dioxide molecules. Here in the animation, I have tried to show you how the dough rises because of the trapment of carbon dioxide molecules. The next product is cheese, where milk is heated first and then lactobacillus is added so that the lactose, which is the milk sugar, is converted to lactic acid, similar as we saw in the formation of yogurt. This lactic acid would ultimately cause the curdling of milk protein. This fats and solid protein curdles are separated from the liquid whey. The mixture of bacteria and fungus is added. Mind it that varying the mixture of bacteria and fungus and pH and temperature will give you different kind of cheese. Salts and preservatives are added then, and the cheese is left to ripen or mature at room temperature. The next product is alcohol production. It is formed by mixing yeast with barley or grapes or any other sugar source. It can be even rice or cereals. The yeast would respire anaerobically, converting sugars to alcohol and carbon dioxide. Changing the sugar source will lead to change or different products being formed. For instance, barley would give you beer, while using grapes will form wine. In spirit-based drinks, for instance whiskey, alcohol can be separated by distillation.